Honors Biology, second quarter review, part 3. Asexual reproduction. The prefix a means without, and sex is having two sources of genetic material in the offspring. So asexual reproduction basically just means one source of genetic material. And we only find simple creatures being able to do this. For example, single-celled eukaryotes reproduce asexually. There's no boy or girl yeast, no boy or girl paramecium or amoebas. They just divide in two, and now you got two of them. Basically, it's a process of mitosis, or making two copies. Simple multicellular creatures sometimes reproduce asexually, such as the hydra. Hydra is a cousin to the jellyfish, about the size of your fingernail, and lives in the water. This hydra here is producing a clone by mitosis. Remember, mitosis makes genetically identical cells, and all these cells here are identical to the cells of the parent hydra. Once this asexual means of reproduction is complete, this bud will pinch off, and now you got two hydras. This is called budding. Basically, it's a asexual reproduction process, producing a clone by mitosis. The advantage of asexual reproduction is that you don't have to find mates. You just make a copy of yourself. And if you're already in a stable environment, you're going to make perfectly or well-adapted clones of yourself that are also going to be able to survive in that environment. The disadvantage, however, is, of course, in a changing environment. So if there's not a lot of variation in your population and the environment changes, that means there's less likelihood the offspring are going to have a survival trait that's going to help them out. Punnett squares. Here we have our pea plants. Remember, Mendel was the father of genetics. Basically, he was the first one to quantify and use the experimental method to figure out the rules on some of the things that we now take for granted with genetics. What we have over here is big P, little p, and remember we have two of every chromosome. Homologous chromosomes have genes for the same trait. The different versions of the genes are called alleles. The dominant allele for a particular trait is represented by a capital letter, and here we have the recessive allele represented by a lowercase letter. So this diploid cell, two sets of chromosomes, you're going to have two copies of a gene, and in this case we have the dominant allele and the recessive allele. Now remember that this diploid cell is going to make haploid gametes. So this parent here can either make gametes with the dominant allele or the recessive allele, and that's represented on a Punnett square. We also have the other parent, and that parent also in this example is big P, little p, having a dominant and recessive allele, and they're going to make gametes by meiosis as well. So the female parts of the plant are going to make either eggs with the dominant allele, big P, or the recessive allele, little p. And then we can do the cross, what happens when the male sperm and the female egg combines together. We could have two dominant alleles packaged up in the zygote, giving us big P for a homozygous dominant. Or we can have a big P packaged up with a little p, making a heterozygote. And we have two ways that we can make a heterozygote, either from the little p in the sperm and the big P in the eggs, or the little p in the eggs and the big P in the sperm. So we have 2 out of 4 chance, or 50% chance, of making a heterozygote. Then the only way we can make a homozygous recessive, 2 little p's, is if both the male and female gametes combine together to provide little p for that zygote. And here we have genotypes and phenotypes. Remember, genotypes are a description of the genes. Phenotypes are the physical characteristics. So what we have here is a 25% chance, 1 out of 4, making big P, homozygous dominant genotype. We have 2 out of 4, or 50% chance, of making a big P, little p heterozygote. And then we have a 25% chance, 1 out of 4, of making a homozygous recessive, little p. That comes out to a 1 to 2 to 1 ratio. As far as phenotypes, remember there's 2 ways to make a purple flower. You can either have 2 of the dominant alleles or a dominant recessive allele. Remember, the recessive allele is hidden if the dominant allele is present. The only way you see the recessive traits is if you get 2 of those alleles. So we have big P, little p, both ways to make purple flowers. So that's basically a 75% chance that in this cross you're going to produce purple flowers in this cross between 2 heterozygotes. And there's only a 25% chance of making white flowers, because there's only a 25% chance of getting little p. So remember, we have 2 ways of making the dominant phenotype, which is the 2 genotypes of big P, homozygous dominant, and big P, little p, the heterozygous. Hetero means other. That means other, one of each allele, basically, big P or little p, and a little p. Homo means the same, so homozygous dominant means 2 of the same allele. big P, big P. If it was homozygous recessive, that would be little p, little p. However, for the dominant phenotype, you have two possible ways of making that. And that makes it more difficult to figure out what the parents are if you have the dominant phenotype. All right, so we have a test cross here, and this is one way we can figure out what the dominant phenotype is. And let's see if you can follow along. We have purple flowers, and we're not sure if this purple flower is big P, or big P, little p. So in a test cross, you cross it with a known recessive phenotype organism, in this case white flowers. There's only one genotype for the recessive phenotype, little p, little p. So if you are big P, if this purple flower that we don't know is big P, and we cross it with a little p, this is our cross. All the children, all the offspring are going to be big P, little p, and that means that you're going to get just purple flowers. What if, however, the purple flower that's unknown is big P, little p, and we cross it with the white flower? When we do this cross, we find out that half the flowers turn out to be purple and half turn out to be white. So if we were trying to figure out what the genotype is for this purple phenotype flower was, we just have to cross it with a recessive phenotype, little p in this case. And if we got any white flowers at all, we know that the parent was big P, little p. However, there is a little bit of a problem. Let's say you get, 5 purple flowers. Is it possible to produce just purple flowers? Get 50% chance, 50%, 50%, 50%, 5 times? Yeah, it's possible. So just getting the dominant trait in the offspring doesn't really help us completely unless we have a large sample size. But if we get any of those white flowers, there's only one genotype that purple flower could have been to get those white flowers, and that's big P, little p. The parent, the purple flower, had to give a little p, and then of course the white flower has only little p's to give. All right, sex-linked traits. These are traits carried on usually the X chromosome. Typically they're recessive. So in this case we have hemophilia represented by the little h. Big H represents the normal allele for clotting factors in people. So this is a female. A female is represented by a circle. A male is represented by a square. That's something that you need to know. Circles females, squares males. Mom, dad. Now if you remember, dads have an XY chromosome, and sex-linked traits are disorders carried on the X chromosome, and males only get one copy of it. Females get 2 copies, and they're kind of lucky in a sense. If the female gets the little h for hemophilia, and hemophilia is a failure of your blood to clot, you can bleed to death from even a small cut. The female also has, if she's a carrier, the normal allele. So if she has the normal allele, that's going to hide the hemophilia allele, and she'll be just fine. She won't have any symptoms. She won't bleed. However, since males only have one X chromosome, if they get the little h, they're going to have the hemophilia. Now this is a normal male represented by a big H. The big H is the normal clotting factor. So let's say we have the male here producing gametes, X and Y. Male's normal, big H. The female produces gametes, XX, and one of them has the normal allele, one has the hemophilia allele, and we do a cross. Well, 25% of the time we get a female that has 2 normal alleles, no problems there. 25% of the time we get a male, Y chromosome, big H, normal allele, so this boy is just fine. 25% of the time, however, we get a girl that has the normal allele and the hemophilia allele. Now remember, hemophilia is recessive, so even though this female has the allele, has the gene, she has the normal gene, so she's going to be just fine. However, she is a carrier, and she could possibly pass it on to future generations. Here's the real problem, though. If you take a look at the son being made, the male only has Y chromosomes to give. If the male gives an X chromosome, it turns out to be a daughter. So if the male gives a Y chromosome and the female gives the hemophilia gene, then you have X hemophilia Y. The reason why males are more likely to get sex-linked traits is because they don't have a second X chromosome that might have the normal allele to hide the hemophilia or other sex-linked disorder alleles. So if this boy gets the hemophilia gene, he has the Y chromosome that doesn't really help him, he's going to have hemophilia. For females, they become carriers. For males, they actually exhibit the disease. And that's unfortunately for guys. All right, Down syndrome. Down syndrome is 3 copies of chromosome number 21. Basically, during meiosis, the chromosomes don't separate out properly, and you get extra chromosome numbers. And you can survive it, but there's a lot of characteristic syndromes that are a problem, and you get a more probability of having Down syndrome when the mother is older. Here we have a karyotype. Remember, a karyotype is a picture of chromosomes. This is a kid with Down syndrome. We have 2 of every chromosome. However, this is a problem right here. We have the 3 chromosome 21, which means the kid's going to have Down syndrome. And we can tell 2 X chromosomes, so this is going to be a girl. Now remember, these are chromosomal disorders. They're not like, you know, sneezing on someone and you're going to get the disease. So it's okay to hug a kid that's has Down syndrome. The chromosome's not going to pop out of all their cells and infect you. We have something called amniocentesis during the 2nd trimester, where the doctor pulls out some of the fluid here, gets some of the cells, and basically does that karyotype and gets a picture of the chromosome, see if there's going to be any problems. Alright, so this is a review of genetic disorders. We're going to talk about this on Monday. And we have chromosomal disorders, basically getting extra copies of chromosomes. The one I want you to focus on is Down syndrome. That's 3 copies of chromosome 21. But there's one called Jacob syndrome, which causes some problems in males, getting extra Y chromosome. Klinefelter's is a problem for males also. Basically, they turn out to be a little more female. Not an intersex. They still have the male organs, but they're sterile. Turner syndrome is 1 X chromosome. And basically that's going to result in a female that's going to get a webbed neck and some other problems. Some recessive genetic disorders carried on the first 22 pairs of chromosomes includes Tay-Sachs, where the brain lipid builds up in your brain cells, nerve cells, and basically the lysosomes don't function correctly. Cystic fibrosis is where you have a protein channel in your lungs not working and other places, and the mucus starts building up. You basically choke on your own snot. Here we have some dominant genetic disorders. You can't hide those. If you have it, you're going to have the disease. And they're pretty serious. Well, one is Huntington's disease causes neural decline by the time you're 30. The reason why these still get passed on is because the people that have Huntington's don't get tested for it, and they pass on the disease before they turn 30. If this was lethal, at 10 years old, that's before the person would reach sexual maturity. It wouldn't be passed on. So the reason why these still persist in our culture is because you have a chance to keep on passing these on, and then the person dies from it. Polydactyly, not quite as serious. Poly means many, dactyly refers to fingers. That's a dominant disease. If you have polydactyly, you're going to have extra fingers, and they can usually just chop them off if you are born this way. Codominant genetic diseases include things like sickle cell. Codominant means that if you have the allele for sickle cell as well as the allele that's normal, you exhibit both of them. And someone with sickle cell produces, or is a heterozygote for sickle cell, will produce the normal red blood cell hemoglobin as well as the misfolded hemoglobin to give themselves sickle cell. And you'll learn about this on Monday. And some sex-linked genetic disorders. They're carried only on the X chromosome. There are some Y chromosome disorders, but most of them are in X because it's a bigger chromosome. We have hemophilia, and you just saw the example in the previous slide. No blood clotting. You can bleed to death very easily. Color blindness, red-green color blindness, is also a sex-linked disorder. And muscular dystrophy, Duchenne muscular dystrophy, loss of muscle control, failure to make a muscle protein to allow you to control those muscles. And all 3 of these are more likely to be found in the males, because if they get the allele for the disorder, they're going to express it because the Y chromosome doesn't help them. Females tend to be carriers because if they get the recessive disorder, all these are recessive, and they have the normal allele hides the disorder, and they don't exhibit any of the symptoms. Here we have sickle cell anemia. Basically, it's just a amino acid sequence in the protein called hemoglobin that results in the blood cell sickling and getting clogged up in the arteries and veins. Very painful, and it's very lethal as well. Many children die by the time they're about 5 without treatment. So basically you have a mutation to DNA that causes misfolded protein that results in a phenotype. The 250,000 hemoglobin per red blood cell causes the whole cell to sickle up like this and get stuck in the different blood vessels. Pedigrees are also called like family trees, and here circles represent females, squares represent males. A line attaching the two, a horizontal line attaching the two, represents a marriage between the two. Any lines coming off the middle that results in other circles and squares are the children of that cross in a pedigree. Now, the other thing I want to point out, in addition to circles being females and squares being males, is that if the circle is shaded, that represents a person with disease or other trait that you're looking for in the pedigree. If the male was shaded, that means the male has the trait. Sometimes you'll see a circle that's half shaded. That means that person's a carrier, which means they do not have the trait, but they carry the disease. Over here we have blood typing. We'll talk more about blood typing fourth quarter, so we're going to skip that for now. Here we have a dihybrid cross, basically two heterozygotes for two traits, and these are the alleles being made. Go ahead and review the process of figuring out the dihybrid cross.